All right, today we're diving into a massive shift that has completely rocked the world of biology. And it's not some newly discovered gene or a weird new creature. It's about the data, an absolute tsunami of it. So picture this. You're a biologist, right? For years, maybe decades, getting data was the hard part. It was scarce. It was precious. But now your lab is just drowning in it. We're talking petabytes of information. So what on earth happened? How did we get here? Okay, let's get into it. For the longest time, getting any kind of biological data was just painstakingly slow and unbelievably expensive. I mean, the first big sequencing projects, they needed huge international teams to even get off the ground. And there was one project in particular that really set the bar for just how much this stuff used to cost. Yeah, look at that number, $2.7 billion. That's what it cost to sequence the very first human genome. It was incredible, for sure, an amazing achievement, but it also cemented this idea that genomic data was this super rare, super precious thing. That was the world we lived in, a world of data scarcity. And then, well, then everything changed. So check out this chart. See that massive nosedive right around 2008? That's not just things getting a little cheaper over time. No, that's a full-blown revolution. This is when next-generation sequencing hit the scene, and the cost just fell off a cliff. What used to take a global effort and billions of dollars suddenly became something a single lab could do. And that tiny trickle of data, it turned into a raging flood real fast. So when you go from a data desert to a data ocean, basically overnight, you can't use the same old map. You need a new way to navigate, right? All of a sudden, the tools you used to use, the skills you had, they just aren't going to cut it anymore. The whole idea of what a lab scale even is, it starts to shift. And I mean, the change is just huge. For those of you who work with data, think about it like this. If you're used to working with nice, clean relational databases, imagine all that data suddenly won't fit. In fact, it's not even a static table anymore. It's a massive, constant stream. You're not wrangling that in Excel on your desktop. No way. You're going to need command line tools, you're going to need remote servers just to have a fighting chance. And this really gets to the heart of it. Getting good at this stuff, it isn't just about learning which buttons to click in some new software. That's kind of like saying being a great molecular biologist is just about being good at pipetting. It misses the whole point. True bioinformatics is so much deeper. It's about understanding the why, the logic, the workflow, the core principles driving the analysis. But, and this is a big but, this whole data revolution has a pretty dangerous flip side. All this new power brings new risks, and a lot of them are hidden. The complexity of how we analyze all this data today, well, it can create a really fragile foundation for our conclusions. And honestly, it's a little worrying. You might think, okay, so what? More data just means we need bigger computers, right? But the real danger isn't about processing power. It's way more subtle than that. It's about whether the scientific conclusions we're building on all this data are actually, reliable. I mean, just think about it for a second. A single result in a paper could depend on a chain of maybe dozens of different programs, each one with its own specific version, its own settings. And these analyses are so complicated that they often get run just once to get the final numbers. What you end up with is this rickety scaffold. Our big, amazing biological conclusions could be resting on something that's fragile, impossible to reproduce, and maybe even flat out wrong. So what do we do about it? How do we build our science on solid rock instead of a rickety scaffold? Well, the answer is we need a new toolkit, a new set of rules and best practices that are actually designed for this new world of massive data. And building that strong foundation, it really starts with simple discipline habits. Things like organizing your projects the same way every time, knowing exactly where your data came from, using tools to check that your files haven't been corrupted, automating stuff so you don't make simple mistakes. These aren't just nice things to do. They are the absolute bedrock of science that you can actually trust and, reproduce. All right, if there is one single idea you take away from this whole thing, let it be this one, the golden rule. You see it on the screen. Don't cut yourself on the computer. And that's because there's a very specific kind of danger in computational work that, well, it catches a lot of people by surprise. And it's this thing called a silent error. See, in a normal lab, a wet lab, when an experiment fails, it's usually pretty obvious, right? The gel is a mess, the cells die, something is clearly wrong. But with a computer,
An error can be completely silent. Your code can run without a single warning, it spits out a result, and that result is just plain wrong, and you'd never know. And that danger is the golden rule. In the lab, a failed experiment is loud. On the computer, a failed analysis can be silent. A bad analysis can spit out results that look totally plausible. If you're not actively checking and double-checking, it's almost impossible to spot the mistake. You have to hunt for the errors, because the computer isn't going to raise its hand and tell you it messed up. Which really leaves us with a huge question for the future of biology. As we're all trying to ride this data tsunami, we have to keep asking ourselves, are the conclusions we're drawing actually solid? Or are we just building bigger and more impressive things on top of that same rickety scaffold, totally blind to all the silent errors hiding underneath?
That's something to think about. If this explainer got you thinking, do us a favor and hit that like button, share it, and subscribe. We'll see you in the next one.